being recorded. I'll have to make sure that I'm polite. Thanks so much for the introduction. In fact, these days I work for Nova from Marklim. It's a reversal of roles. Nova used to work with me, and now in my retirement, I'm Nova's voluntary shore assistant. So what I'm going to talk about this evening is I'm just going to talk about what limpets do on rocky shores, and this is based on 45 years work that I've been doing over the last 45 years really. I started as a very scruffy undergraduate in Liverpool in 1975, being interested in limpets. I actually come from North Devon, from near Braunton, but I wanted to get away from Devon, so I went as far away as possible and did my education at Liverpool and the Isle of Man. And I went on an inspirational seaweed field course, an algology field course in Easter 1975. And the lecturer in charge got me looking at what was growing on limpet shells and comparing it with the surrounding area on the grounds that limpets aren't very good at grazing on themselves, and getting an idea of what's on the shells gives you some idea of what there might be if the limpets weren't grazing so effectively. And that got me interested in rocky shores. So I am a fairly unashamed lover of limpets. So why do people like me study rocky shores? I do it for fun now, but in my professional life before I retired, I did it because they were such a neat experimental system. They're easy to sample. You can take photographs, you can count quadrats. Most importantly, all the organisms on the shore compete for an easily measured resource space. And most of them have short lifespans and they're manageable sizes, and a lot of them don't move around very much. So you've got plants and animals competing for the same space, and any predators and grazers tend to be sedentary and don't move around very much. They're ideal for experiments. And also there's two very clear, sharp environmental gradients on rocky shores. From the sea to the land up the shore, a vertical gradient, and a horizontal gradient from wave-exposed headlands round to sheltered bays. And that makes it experimentally manipulate things. There's also very few taxonomic problems. It's quite easy to identify most things accurately, and all of the above make doing experiments easy. You can move things around, you can exclude them, you can remove them, you can thin things, and you can do all this work with an adjacent control area next door, a reference area, so you can see what your experiments are doing to the test system that you're playing with. And I think one thing I really want to emphasize is the very rapid response times. If you want to have a look at the effect of removing grazes on native vegetation, you have to wait 400 or 500 years before you get a climax oak wood. But on rocky shores, you get things getting back to normal quite quickly. It might be helpful if somebody muted their microphone. There's a bit of background noise going on, which is feeding through, but it's not me. So perhaps if everyone can mute their microphones, it might help. So I suppose you can sort of consider rocky shores as being the lab rat of ecology. And lots of contributions to general ecological theory have come through their study by lots of people all around the world. And if you're nerdy and you're interested in that kind of stuff, myself and some colleagues published a paper recently in the 100th issue of the Journal of the Marine Biological Association, just giving an idea about how useful rocky shores are as very tractable test systems for experimental ecology. And over the years, I've published a few books on the subject. Some of them are really cheap now because they're out of print, so you can get Intertidal ecology and you can get the Rocky Shore ID guide for next to nothing on eBay. So you know, probably worth buying if you're prepared to lash out £5 in the postage. And more recently, we summarised things in an edited book in 2019, which includes an overview of what shapes rocky shores throughout the world. But there's the opening chapters on the Northeast Atlantic. So what I'm going to talk about is a little bit about grazing mechanisms, a little bit about what limpets feed on, talk about some of the classic early work that was done on the Isle of Man by one of lecturers that used to lecture me when I was in my 20s and he was my age and in his mid-60s. I'm going to talk about some experiments that my group have done over the years on grazing and how limpet grazing influences rocky shore community. Some of this is on a European scale. I'm going to talk a little bit about how grazing determines vertical patterns on the shore, zonation, and then talk about what happens when lots of limpet gets killed, using work that Alan and Eve Southwood did, and which I've continued since about 1980, on the aftermath of the Torrey Canyon oil spill. And then I'm going to finish up with why seaweeds dominate on sheltered shores and pose a couple of questions, which perhaps some of you might be able to go out and help me look at in more detail. And also a colleague from Brazil who's going to come over and hopefully work on this next year. So the vertical gradient from totally marine conditions to totally terrestrial conditions is very sharp indeed. And most of the animals and plants you get on the seashore are marine evolutionary affinity. You get very, very few species that are terrestrial in origin. There's, I think, one species of spider, a pseudoscorpion, a few mites, lichens possibly. But most of the animals and plants are marine species colonizing the intertidal. So for them, there's a unidirectional stress gradient going up the shore as you go from low levels to high levels. And there's my mate Louise demonstrating zonation on a rocky shore somewhere in North Cornwall, with mussels at the bottom and barnacles at the top. The other gradient is between exposed headlands, which are dominated by lots of barnacles and limpets and occasionally mussels, into shelter, which tend in Britain to be dominated by dense beds of seaweeds. This is the egg rack ascophyllum, completely coating a sheltered rocky shore on the Isle of Man, with King William's College in the background there. So I want to try to get over to you there's a dynamic balance between grazers in exposed conditions and between macroalgal seaweed domination in more sheltered conditions. And though this is modulated by wave action, what actually tends to directly set patterns is largely grazing activities and limpets. Just to think about what happens as you go from a sheltered bay out to a headland, there's various benefits with greater wave exposure. Things get wetted by waves, particularly high on the shore. There's a lot more plankton food for filter feeders like barnacles. There's often a greater supply of larvae to exposed headlands being taken there by water movements and currents. And there's a lot more oxygen and nutrients. As you go into shelter, there's things can get a little bit easier in terms of dislodgement. There's quite a bit of risk of being dislodged on more exposed shores.
I can't see what they're talking about at all. Well, it's gone now. I can't see anything.
Hello, I can see you.
Can we mute, please? Can we mute?
Yeah. Thank you. Hi there, nice dogs. As you go from sheltered condition out to more exposed conditions, it gets more difficult for plants and animals like barnacles to stick on the rock. So there's problems with settlement and there's a greater risk of dislodgement. Similarly with mobile animals, you're more likely to get knocked off by waves if you're foraging. On the other hand, as you go into more sheltered conditions, there's a lot more silt. Things get smothered by silt, and this can interfere with respiration in animals. Silt can clog up gills, and it can also cover seaweeds and prevent photosynthesis. It can stop light getting through. So let's get back to limpets. What do? Well, limpets have a home. They go off and forage. They lay down a mucus trail on the rock. This is an area with quite a bit of silt being deposited. You can see individual limpet mucus trails there. They depart from their homes rapidly on horizontal surfaces when the tide comes in, and then they come back down again, back to their home. Just
you can see somebody doing something.
And they do feeding excursions all the way on those particular trips. They actually follow the mucus trail.
Yeah, that's just him.
Sorry, I'm picking somebody up in the background again. They forage, they actually follow their home mucus trails back to their homes. They use it to home. Limpets have really hard teeth, some of the hardest teeth in the animal kingdom, and they feed with something called a radula. And it's a bit like a long ribbon where the teeth at the front are being worn down as they're used for feeding, and they're getting replaced from the back. And this is a scanning electron micrograph of the teeth of the limpet. These very, very strong teeth, toughened by iron oxide. And here you can see where they've been scraping away on the rock surface. These are individual limpets.
I thought I was unmuted. I was muted.
So that's how they feed. And in fact, limpets not only feed on what's on the surface, but they probably eat a little bit of the plate itself. They actually grind away at the rock surface, particularly with softer rocks. And if we compare the feeding marks left by different...
Oh, I can't find anything.
Can the people that are talking please mute?
Yeah, hello there, it's Mike, admin tonight. Can please everybody mute their microphones because it's spoiling the talk. Can you just check, please, everybody, and turn off? Thank you.
Okay, thanks very much. So if you've got, these are different types of snails and limpets on the shore, and each of them have got different types of radulae teeth. And top shells have a very fine radula with fine teeth, which it uses to brush up the algae on the rock. And Littorina littorea have sort of like a Swiss Army penknife type radula, where they can use it in lots of different ways to bite, to rake. And these are marks left by Littorina, by winkles on glass slides, which have been coated with soot. And these are actually the tooth marks. These are the tooth marks left by Ocelinus. And these are limpets that I conned into feeding on a plastic petri dish by rubbing some seaweed on it. And these are the tooth marks of Patella vulgata. And these are the tooth marks of Patella, what used to be called aspera, now called ulyssiponensis. And these are very deep gouges into the plastic petri dish. They do this on the shore as well. They actually gouge out the calcareous algae that they live on, the pink encrusting algae that they live on. Now, when you go on a shore, sometimes it looks bare, but actually it's not. And there's this amazing invisible lawn of microalgae, of diatoms, various filamentous blue-green bacteria or blue-green algae. These are diatoms, all in a matrix of mucus and various polysaccharides. You also, up here underneath Mike Poulston, you get, this is a picture of some germinating algal sporelings or germlings. So this is very much like a grazed field. The sheep are eating the grass as quickly as the grass can grow. It's a bit like you mowing your lawn. And the limpet grazing is keeping this lawn of macroalgae. They're feeding on them. You find all these diatoms in their guts, and it's a bit like gruel. If they get a chance, they'll eat something bigger, but a lot of the time on exposed rocky shores where there's no big seaweeds, it's this gruel that they're feeding on. And there's some evidence that actually they retrace their mucus steps and they actually feed on phytoplankton that deposit out of the water and stick onto the mucus. So these biofilms are really important, but it's limpet grazing of what's going on in the biofilm, particularly baby algae, that determines the amount of vegetation on the seashore. So how can you measure grazing intensity and pressure? Well, one of my very imaginative PhD students, a guy called Richard Thompson, who's now very famous as a fellow of the Royal Society, got an OBE for his fantastic work on plastic pollution, was very cunning when he was a student, and he came up with a way of measuring how limpets feed. And this involved putting dental wax in screw covers, which he then inserted into the rock. And then you can take these out afterwards, you can actually see the feeding marks left by the limpets. And if you have an array of these, you can measure how extensive and intensive the limpet grazing is. So this is a very good indirect way of measuring grazing activity with some classic low-tech approaches. I don't think you can make limpet dentures out of this dental ones though. So intertidal biofilms, very important. A lot of coastal primary productivity, which goes through limpets. Probably most of the limpet energy comes from biofilms. And this is a picture from Mike. It's also where things settle. These are settling barnacle larvae. That's a cypress larvae, which is about to metamorphose in a day's time into that, in 2 days' time that, and in about 2 years' time becoming this. But what you can also see in this micrograph is these filaments here are the early stages of macroalgae, which are just beginning to sprout and grow on the rocks. And a bit later they get bigger. This is probably a baby fucus or rack plant. And if it hadn't been chipped off the rock for examination under the electron microscope, it probably would have got eaten by a limpet in 2 or 3 tides' time. Now, one of my old lecturers, Norman Jones, who did some very pioneering work, and up until the 1930s, all explanations of patterns on the seashore were very much driven by physical explanations. This light coloration here is a barnacle-dominated shore, and the explanation in the 1930s and early 40s would have been that it's covered with barnacles because it's too rough for seaweeds to occur there. But Norman Jones removed all the limpets in a strip down the shore, and you can see this stripe here is lots and lots of seaweeds which have grown in the absence of the limpet grazing pressure. Being the late 1940s and there being an airbase on the Isle of Man, he managed to get a reconnaissance mosquito to come and take a picture of his 10-metre wide limpet exclusion strip. This is an exciting bit of work. He observed that he actually removed a lot of limpets for measurements, and he observed that a lot of seaweeds grew there. So he extended the strip all the way down the shore. He buried it in the a really top journal, the Proceedings and Transactions of the Liverpool Biological Society. Now, some 30 years later, I started my PhD on the Isle of Man looking at grazing interactions. And I read these papers and I was a bit skeptical about it. So I repeated the experiment. I removed limpets in a 2-metre by 10-metre strip down this ledge, completely covered by barnacles. And within 2 months, I got lots of green algae. And 9 months later, I managed to induce, being in the punk era of the 1970s, this fabulous mohican of fucoids all the way down the shore. And then a little bit later, the limpets on either side migrated under the fucus clump. And as a consequence, there were less limpets here and more seaweeds occurred either side. Now, had I continued with this methodology, I would have soon run out of rocky shore on the Isle of Man. But about the same time as this, my supervisor and I came up with a wheeze, which was to actually drive power drills off scuba cylinders. Remember, in the 1970s, cordless drills hadn't been invented, and the health and safety problems of taking electric generators on the shore, as well as affordability, was somewhat too much for the lab we were at. So we invented a way of drilling holes in rock, and we were able to do much more sensible experiments. So I started building little fences to keep limpets out. So this is some chicken wire, this is a 1-metre area, this is the treatment, and here is the control area. And you can see that in the treatment, there's lots and lots of fucus, and in the control area, there's very little growing. So this is the treatment, some 9 months later. This is a control, very little seaweeds, a few here which have escaped naturally, a few fucus growing on the shells of limpets, because limpets aren't very good at grazing on themselves, but much, much more in the treatment than in the control area. So I did various other experiments. This is one which I set up in the spring, and there were green algae first, then followed by these brown seaweeds, by these fucoids afterwards, but all pretty much showing that, that limpets were having a really strong effect on the seaweeds. Now, some of this work was criticised because of lack of replication, but sometime later we had a second bite of the cherry when we got some European funding to try to investigate what was structuring rocky shores throughout Europe. And with colleagues in Liverpool, I'd moved to Southampton by then, but we were working mainly around Plymouth, in northern Spain, in central and southern Portugal. We were able to look at the effects of patella grazing, limpet grazing throughout Europe using exclusion cages and fences. So those are the various departments involved. And what we were trying to do, we were trying to look at how grazing pressure affected recruitment of seaweeds, particularly brown seaweeds. But there were lots of other variables going on. We had to make sure that we did it in areas with similar barnacle cover. We tried to avoid too many cracks and crevices, so we tried to choose shores with smooth rock, which we managed to do. We tended to make sure these were gently sloping seaward, and we tried to avoid any microclimate effects, so we tried to do this on open rock, not in any shade. So we sort of basically kept these variables constant so we could explore the effect of grazing on seaweed recruitment. So this is one of the exclusion areas, and you can see the fence there, and this is just before the limpets were going to be removed. And we set this up in lots of different places, 5 countries. In each country we did 2 shores. We set these experiments off in the summer and the winter because we thought in the winter there would be less harsh conditions for seaweeds, particularly in the south, and they might do better. And we did this on a couple of random dates in summer and a couple of random dates in winter. And it was a very complicated experimental design with a very complicated statistical model. Unfortunately, this is what you have to do to get stuff published these days. So that's the exclusion. That's the control, where we left bits of the fence in place but allowed limpets access just to test if there was any enhancement of algal cover in the corners due to the fence itself, which didn't occur. And then we had some unfenced controls where we did nothing as a completely blank control. Fortunately, after getting all this European money, we got some very large effects. So this is all the green algae growing after 3 months on a site on the Isle of Man. This is the fucoid canopy growing a few months later in the same area, all these brown seaweeds. And we were able to compare what was going on throughout Europe from the north down to the south. And this is the maximum cover of big brown seaweeds, of fucoids, of racks. And on the Isle of Man, there was a big effect. There was some that naturally escaped in the controls. In Devon, South Devon, there was an effect, very little growing in the controls. Similar in Spain, but a bit more in the winter. And by the time we got to Portugal, there were hardly any brown algae at all. But if we looked at all species of algae, the maximum cover of anything, including various small greens and reds and things, even in the south, in Portugal, limpets, this is the difference between the treatment and the controls. The limpets were having a very strong grazing effect and actually preventing ephemeral green algae from growing on the shore, even in Portugal. So grazing is really important. It's just different. It controls different species in different parts of Europe. So this is the canopy cover. Again, a much stronger effect in the Isle of Man, and then southwest England and northern Spain, moderately similar, and then very little fucus at all in central Portugal, and some bonsai fucoids, very, very small stunted plants in southern Portugal. So big difference between treatments and controls. Some effects of season and location, particularly the strong north-south gradient, and also more seaweeds colonising quicker in the winter in the south when it was better conditions for seaweeds than in the harsh summer. And there were some differences in the sequences of colonisation in different places. So in the Isle of Man, it tended to go either to greens and then brown seaweeds, or sometimes straight to brown seaweeds. In Devon, it generally went to greens and then fucoids and occasionally some crusts and some turf seaweeds. In northern Spain, similar patterns to South Devon. And in Portugal, it went to greens and occasionally to crusts and tiny little fucoids, the only species which is able to survive that far south in Portugal. And we did lots of complicated statistics, but that's boring. So let's just have a little bit more of a closer look about what went on in the British Isles. And there was a much lower effect of limpet removal in southwest England. And in the Isle of Man, this is a big word, deterministic. It just means if you do something, you know what the response is going to be. It's pretty obvious if you remove limpets there, you consistently get lots and lots of seaweeds. But in southwest England, it's much more stochastic. That's a fancy word for it being a bit iffy. Sometimes it works, sometimes it doesn't work. And that's very much due to the fact that perhaps the grazing pressure is not quite as important as other factors. And if we look at this in a bit more detail, we can see that the summer exclusions, which then led to stuff growing in the summer, was quite rapid in the Isle of Man and much slower in southwest England. In the winter, it was also faster in the Isle of Man, but slightly faster on the winter initiations, which means that these were growing the following spring in southwest England. And if we look at how often each of these outcomes occur, then we can get some idea of of how likely it is to occur. So on most occasions when we did experiments on the Isle of Man, we got 100% cover of brown seaweeds, of fucoids, of racks. When we did it in southwest England, it was much, much more smeared across the whole range of experiments. So just to summarise, grazing clearly controls seaweeds on barnacle-dominated shores throughout Europe. Limpets are something that can be called a keystone species. They have a really huge effect on completely structuring rocky shore communities. The effects are greater and they're much more predictable further north. But physical factors, season of when the limpets are removed, and also latitude influence the colonisation rates. And there was an awful lot of variation in the early colonisers, which is very much due to chance, but it became more uniform later on during the colonisation or successional sequence. So why are there less macroalgae on shores in southern Europe? Well, firstly, there's far more diversity of grazers. There's a lot more species of limpets. There's also lots more snails, top shells, various other grazing species, even herbivorous fish. There's probably greater grazing pressure, and we actually measured this on a European scale using these wax discs, and we actually showed that there was grazing pressure throughout the year in the south, whereas it was much more seasonal in the north. And there's also an increase in physical stresses. Clearly, it's very hot and dry, which is not good if you're a germinating seaweed plant. So there's a couple of potential explanations. One is that limpets are doing everything, and they're so effective they completely eliminate fucoids. The other competing idea is that fucoids are not adapted to increased physical stress in southern Europe. We actually tested this. Stuart Jenkins and I had a student who did work in North Wales and in Portugal, and he actually showed it's a combination of the two. That further south in Europe, there's a lot of stress, particularly on early germinating seaweeds. There's also less supply of propagules, there's less eggs and sperm being produced by the fucoids, and that grazing also is involved and determines the actual probability, the likelihood of these seaweeds escaping, of fucoids, the brown seaweeds escaping. Now let's just go back and run a health check on whether physical factors are completely unimportant. And we had a great opportunity in another European project, working with some Swedish colleagues, where we're actually looking at sustainable design of sea defences. But we also did quite a bit of ecology on the side. And we were using this test system in Bognor Regis. I don't know why you bothered to defend Bognor Regis, to be honest. I could quote George V here. And we actually compared the seaweeds growing on the seaward side of these sea defences with the landward side. And actually there's a lot more fucus on the sheltered side, not surprising. Barnacles are about the same, and there's far more limpets on the seaward side than the landward side, and there's a bit more ephemeral algae green sea lettuce and things of that nature on the landward side. So we measured wave action using a really old-fashioned technique, which is how fast plaster of Paris blocks dissolve. Obviously, more wave action, they'll dissolve quicker. And we also used some fancy gadgets that the Swedes had. This was the English improvisation. This was the Swedish technology coming in, actually measuring the wave forces. And the wave forces were much greater on the outside of the breakwater than the inside of the breakwater, which is not surprising because it's a breakwater. The next thing we did was to remove limpets on both sides of the breakwater, and we showed that if we remove limpets on the seaward side, there was quite rapid growth of fucoids, of brown seaweeds, and if we thinned them, there was also an increase. And the same thing happened actually with the few limpets which were available on the landward side. It also led to an increase in the amount of cover, but there was more cover there anyway. So we got confirmation that limpet grazing was important here. We then measured, or rather my Swedish colleagues measured, the critical breaking point of seaweeds of different sizes under different wave flow, and then we were able to work out the probability of dislodgement of plants of different sizes on the landward and seaward sides. And basically anything bigger than 20 centimetres has a snowball chance in hell of surviving on the wave exposed outside breakwater, so only small plants can survive, and then at very low levels. Whereas on the seaward side, there's very good survival of big plants. So at the end of all this, we could work out, and we also measured the decline of seaweeds over time. They tend to get much smaller because all the big ones get pulled off on the, get plucked off. You have to be careful when you're saying fucoids being plucked off on the seaward side compared with the landward side. And the decline in numbers was much more rapid on the seaward side than the landward side. And those that did survive tend to grow and get bigger. So we can see the survival was much less there. So to summarise then, basically limpet grazing prevents establishment of recruiting fucoids, actually stops the baby seaweeds from growing, but it's actually wave action which reduces the persistence, how long these plants last once they've managed to escape limpet grazing. So on a highly exposed shore, even if you were to exclude limpets, the plants might only last a year or so, whereas on the sheltered shore, they tend to persist for a long, long time, and they don't get dislodged by waves. So grazing prevents establishment, but wave action reduces persistence. The last thing I want to talk about, second to last thing I want to talk about is vertical zonation. Is grazing involved in this? And with another Portuguese student, Diana Boaventura, we did some really neat experiments in Portugal compared to the UK. This was done at Swanage and also at Haybrook Brae in South Devon. We thought that if we did this in Portugal, we wouldn't be able to get any extension of these seaweeds up the shore. Whereas I'd already done some preliminary experiments at Swanage and managed to reduce this algal turf higher up. So we repeated it properly with lots of replicates and another replicate shore, 2 places. And we managed to get this algal turf to extend higher up the shore in the UK, but we also managed to get it to extend higher up the shore in Portugal. So if you read all the textbooks in the 60s and 70s, you'd read that the upper limits of species on rocky shores are primarily set by physical factors. But this actually shows that grazing, limpet grazing, is stopping these seaweeds from extending higher up the shore at this layer. And on a shore like this, there's a really sharp line. And that's because there's a flip between the low shore area where the seaweeds win and can grow faster than the limpets, and the mid-shore region where the limpets are grazing faster than the seaweeds can grow. And also, obviously, the growth rates are a bit slower here as it gets higher up the shore and they get a bit more stressed by physical environments. So there's a flip between the limpets winning and the seaweeds winning. And we also removed the algal turf here, we managed to get the limpets to extend lower down the shore. But unless we kept removing the algal turf, they then eventually got swamped by the seaweeds. So seaweeds are prevented from extending up the shore by grazing, not by desiccation, a physical factor. So what happens when all the limpets get killed? Well, the best example of this, unfortunately, was when the Torrey Canyon ran aground in March 1967 and was studied by Eve Alan Southwood from the MBA. And as happens in all disasters in the UK, you send the army in. These guys here are having a competition in terms of how much they can spray detergent up the wall. Lots and lots of detergent dispersant was sprayed to clear up the oil ahead of the holiday season. But this made things much worse. In Cornwall, for instance, 14,000 tonnes of oil came on shore. This was crude oil, not particularly toxic, onto which 10,000 tonnes of highly toxic dispersant was poured. So this was made worse by inappropriate treatment. And these dispersants were really industrial cleaning fluids. They were mainly organic solvents with a bit of palm oil to stabilise them. They were more toxic than crude oil, and they killed all the grazes, particularly limpets. And as we've seen from earlier in the talk, limpets are really important in controlling algal vegetation. So these are pictures taken by Alan and Eve Southwood. This was a year after the spill, and this was previously a barnacle-dominated shore, now completely covered by large numbers of brown seaweeds. And this persisted for the next 2 or 3 years. You can see the shore's completely dominated by brown seaweeds. Now, these seaweeds are a fantastic nursery ground for baby limpets. So limpets that have reproduced in unpolluted areas, got here by larval transport, settled and did really well. So massive numbers of limpets started recruiting underneath this fucus canopy. And then subsequently, the shore went very bare. And this was because these large numbers of limpets were actually feeding on the seaweeds and grazing them down. So that's the first 10 years or so of the sequence, lots of brown seaweeds, and then from about 73 onwards, the shore getting very, very bare. And this is subsequently the shore getting back to normal. I first started working here in 1980, and Alan Southwood said to me, "Do you think the shore's normal?" I said, "No, I don't think so." So he said, "We'd better keep on looking at it then." And I was down there earlier this year. We're still doing it now, 52 years later. So the shore started to get back to normal about 82, 83, and that's in 87. That's in 97. But if we look in more detail at one of the patches on the shore here, you can see what it was like at time 0. Lots and lots of barnacles and a few fucoids in natural patches. This is sort of natural conditions. Then after a year, lots of green seaweeds and brown seaweeds, and then complete proliferation of brown seaweeds, and then beginning to get bare as all the limpets, which are settled underneath here, are beginning to grow and munch on the seaweeds. And then it getting very bare indeed in 74, 75, 76. So if we have a look at a graph, some greens, some browns, a bare period, then another green period. That's when I started getting involved in 1980. And you can see this massive recruitment of limpets, and then they then died. And probably because they died, there was this second outburst of seaweeds. And then the population's beginning to stabilise, but quite slow recovery of Patella depressa. And the limpets which recruited in such massive numbers underneath the seaweeds form these dense populations. And normally limpets home, but because they were starving, they migrated across the shore like lemmings in these massive fronts, ate all the seaweeds and eventually started to die, which is what this massive decrease is. Now, there's 2 species of limpet here. There's the northern species, Patella vulgata, and there's a southern species, Patella depressa. And there's been quite slow recovery of Patella depressa. Why was this? Well, when the Torrey Canyon went down, it was during a cold period, which started in about 62, 63. And actually the 60s, 70s and early 80s were quite a cold period until things started warming up in the late 1980s. So not very good for Patella depressa. And this is a warm water species. And when I first came to Plymouth in 1980, I was going to work on competition between these 2 species. And I read in the literature there were shores near Plymouth with 50-50 populations, ideal for doing competition experiments. And when I got out to look at these shores, I found that the Patella depressa were much, much rarer in 1980, 81, 82 than they had been in the warm 1950s. This started my interest in climate change. So when things got warm again, Patella depressa started to appear. But the other thing that we noticed was that when there was lots of algal cover, Patella depressa did less well. But when there were bare periods, Patella depressa did better. And another student, Pip Moore, did some nice work showing that Patella vulgata does really well under fucus, but Patella depressa shuns fucus clumps. And in subsequent years, you can see that when there was bare periods, this isn't very much fucus, Patella depressa does well, and then Patella vulgata does better when there's more fucus because they aggregate and they recruit underneath these fucus clumps. And of course, we continue this, and this is in 2016. You can see the fucus. This is the oil spill, and probably natural cycles. This is a natural recruitment of fucus. It happens in some years. No pollution, just a lot of seaweed recruitment, and then these various cycles. And when there's little fucus, lots of Patella depressa, and when there's lots of fucus, more Patella vulgata, and a bit of a peak here with Patella depressa. So fluctuations going on here. And on the 50th anniversary of the Torrey Canyon oil spill, this got out on the shore, and there were lots and lots of fucus, perhaps in celebration of the seaweeds coming back big time. And here you can see going up again. And in the last couple of years, I haven't graphed this up recently, it started to drop back off again. Now, there was one shore, Grazing, which was not sprayed by dispersant because of the seals. The National Trust didn't allow them to do this. And 2 to 3 years later, the oil naturally disappeared. When the toxic stuff evaporated off, the limpets actually helped clean up a bit because, you know, crude oil is an organic substance. It's got a lot of energy in it. That's why we burn it. And the shore got back to normal in 2 to 3 years. So without spraying, recovery time is 2 to 3 years. With spraying, maybe 10, maybe 12 years in extreme cases because of killing off all the limpets. So just to conclude again, why are shelter shores dominated by ascophyllum? Well, Stuart Jenkins did this work for his PhD, and he experimentally manipulated both ascophyllum, egg rack, canopy, and limpets on the shore. And he did this by having various combinations with and without canopy, with and without grazes, with canopy, with grazes, with canopy, without grazes. And in big 2 by 1 metre plots on the shore here on the Isle of Man, and this is one of the canopy plots beginning to show green algae coming in soon after removal. And he found that the major effect on ephemeral algae, these are what used to be called entomorpha, now called ulva, was mainly the canopy. There was perhaps a little less in the areas where the canopy and the limpets had been removed, but there weren't many limpets there anyway. So the canopy was having a really strong effect on ephemeral algae and also on less competitive seaweeds, fucoids, vesiculosis, and the serrated rack and the bladdered rack. And this is an area underneath the canopy. There's a natural few limpets and a natural clearing being hemmed in by all these turf-forming algae. And this is an area where the limpets have been removed under the canopy, and this bare space is being filled in by encroaching algae of various kinds. And if we look at the amount of bare substrate, either bare rock or this pink crust, where the canopy was removed and the limpets were present, the actual amount of bare space went up as the limpets were beginning to win and keep it clear. This is a control, nothing much happened. Where the canopy was removed and the limpets were also removed, you get a lot of cover going down. There's lots of ephemeral algae cover it, and lots of canopy species coming in and sweeping around and stopping ephemeral algae, and then beginning to go down again as more seaweeds colonise. And if the canopy was present and the limpets removed, just a slight increase in bare rock underneath the canopy as the few limpets were there, beginning to expand their clearings. So ascophyllum, certainly on the Isle of Man, prevents or limits development of other seaweed species. And I think it also facilitates the growth of these red algal turf. And I think this is detrimental to the limpet population. It limits the amount of space for them. And also these algal turfs trap silt, which limpets don't like. Now, Stuart got a job at the MBA, and I managed to persuade him, because I was the director, that he should go back and revisit his PhD. And he actually had a look at these experiments some long time later. And these are the controls where the canopy was left in place, and these were the canopies removed. Very, very slow recovery of ascophyllum, a long phase of domination by the seaweeds, but beginning to drop off as the ascophyllum came back again. But the amount of bare rock was much, much greater. Where the canopy had been removed, the limpets were beginning to do really well in the absence of canopy. Numbers were increasing and they were keeping the rock clear. So this was happening. The limpets were beginning to win once the ascophyllum, the egg rack, had gone. And this was the number of limpets per square metre in canopy removed versus canopy present plots. So clearly there's a very strong indirect effect of the ascophyllum on the canopy. So ascophyllum helps algal turf traps silt, and probably indirectly limits limpet populations. Limpets don't like silt. Also, we think crab predation could probably be more important on sheltered shores. Crabs eat limpets, particularly baby limpets. So on this wave exposure gradient, limpets win in more exposed conditions, but macroalgae limit limpet abundance in more sheltered conditions, but probably through indirect effects. So there's this dynamic balance, as I said at the beginning, between grazing and seaweeds moderated by wave action. But Toby's flyer is interesting because this is at Watermouth Bay, quite a sheltered site, and lots of ascophyllum here is being munched away by limpets. They're beginning to win. And just round the corner from where I live in Newton Ferrers, this is a very sheltered site. It should be covered by ascophyllum like up here. But the limpets are winning here, beginning to get these really, really barren grounds taking over. And I've also seen this in Salcombe. I've also seen it further south in France with Nova, at the towards the southern limit of ascophyllum in Europe, where ascophyllum's having a bad time and the limpets are winning. So I asked the South Devon group and any North Devon people to get out there on sheltered places like up the Salcombe estuary, where you'd expect there to be lots of ascophyllum, and see if the limpets are beginning to win. And I wonder why. And maybe that might be the first question I might get as I'm just about to finish. And if any of you can be bothered, there's loads of references, and as this talk's being recorded, you can freeze frame this, which summarise a lot of the work that's been done there. Thanks very much for your attention. And I'm sorry a dog just barked downstairs in our house at the end. Thanks very much indeed. Cheers, and questions, please.
Yeah, thanks very much, Steve. That was